Welcome to homework.
Okay, when we look at the interactions between these different side chains, we have to consider the actual chemical makeup of the side chain to see how how they will interact together. So when we first look at cysteine and cysteine, right? Cysteine and cysteine actually forms a link between the sulfurs, so it's a disulfide linkage. So when a bond forms between two nonmetals like sulfur, it's a covalent bond or a covalent interaction, right? So that's our cysteine and cysteine, right? For glutamine and serine, right, that they have nitrogen and hydrogen bonds with oxygen and hydrogen bonds. And we know that those are very polar bonds. When those can interact, they actually have a hydrogen bonding force, right? So due to those nitrogen, hydrogen, and oxygen, hydrogen bonds, right, the intermolecular force that forms then between the side chains is going to be the hydrogen bonding IMF. Okay. For glutamic acid and lysine, that is actually going to be ionic, right? So we have a few examples, right? So things like the lysine, which has that NH2 group, right? And the glutamic acid, which has the carboxylic acid group. When those actually interact together, they are able to bond in an ionic bond. And last, we have proline and leucine. So proline and leucine all have are both nonpolar groups, right? So when groups are nonpolar, the strongest force that can possibly form is London dispersion. So that's our pretty weak IMF. Oop, that's wrong. Let's try that again. London dispersion. That's our weak intermolecular force. So if we were to compare all of these, that would be the weakest of all of our forces present.